Alright, this video is going to be about section 2 of chapter 4. We're going to be talking about prokaryotic cells. Now, just to break down the word prokaryotic. Pro means before, and karyotic or karyote comes from the Greek or Latin, I can't remember which one, Greek or Latin base of kernel. So this means that prokaryotic cells came before cells with a nucleus. So prokaryotic means no nucleus. And that's one of the defining characteristics. They lack a membrane-bound nucleus. Structurally, they are smaller and more simple than eukaryotic. The prefix "eu" means true. Karyote means kernel or nucleus, so they have a true nucleus. Prokaryotic cells can be divided into two taxonomic domains, bacteria and archaea. Bacteria cause disease, but they're also environmentally important. They're used to break down matter, to recycle things. We even need bacteria in our bodies. We have the E. coli bacteria in our colon right now helping us digest lunch or whatever meal we just ate. Right? They are very, very important. And they can be very useful in manufacturing products and drugs. Archaea, the other type of prokaryotic cell, live in very extreme habitats like ocean floor vents and areas like that. Okay, they are structurally similar but biochemically different. So from the outside they look the same, but they got different stuff going on the inside. They're extremely small. They're usually 1.1 to 1.5 micrometers wide and about 2 to 6 micrometers long. They occur in 3 different shapes: spherical coccus, rod-shaped bacillus, and spiral spirillum or spirochete. If it's rigid, it's spirillum, and if it's flexible, spirochete. The cell envelope, the outer part of the prokaryotic cell, includes 3 things: the plasma membrane, which is made of those phospholipids that we talked about last chapter, the cell wall, which is made up of peptoglycan. This also contains other molecules, but mainly strengthened by peptoglycan. Glycan meaning a sugar, pepto meaning like polypeptide, so protein, sugar, and a layer of polysaccharides on the outside of the cell wall. Okay, it's very well organized and it's very resistant to removal. Okay, this is also known as the capsule. All right, and it's like the outside layer of the cell of the prokaryote that protects it. Here we have the different shapes of prokaryotes. So you'll notice the spherical, the rod-shaped, and the spiral. Okay. Here we have the plasma membrane. We have proteins that are embedded. Okay, those help with getting things through the membrane and with signaling. And it's also made up of phospholipids, mainly out of phospholipids. It's a bilayer, meaning that there's 2 individual layers of phospholipids that make it up. The polar heads are on the outside, and the nonpolar or hydrophobic tails are on the inside. Remember, fatty acids don't like water. They're nonpolar. They don't want to mix with water. That's why they're oriented into the middle. Here we have the generalized structure of a prokaryotic cell. We're going to go into more detail about these structures in the following slides. The first we're going to talk about is the cytoplasm. It's a semi-solid solution that is encased by the plasma membrane. It contains water, organic materials, inorganic materials, and enzymes. I kind of compare it to Jell-O. Right? In the cytoplasm is the nucleoid region. In prokaryotic cells, they don't have DNA like you and I do. They have one long circular strand of DNA, and it's kind of all tangled up in the middle. There's extra little bits of DNA called plasmids that can be traded between prokaryotic cells. Also found in the cytoplasm are ribosomes, and that is where proteins are made. Proteins are important for all living things to have. Now, an important thing to note about ribosomes is they don't have a membrane. They are basically RNA and protein that are stuck together. Prokaryotic cells also have external structures, flagella, which are like tails that help them move through the water environment that they live in. Fimbriae are small bristle-like fibers that sprout from the cell surface, and conjugation pili are rigid tubular structures that pass DNA from one cell to another. That's how these plasmids are exchanged from one prokaryotic cell to another. Sometimes they're beneficial, sometimes they're not beneficial, sometimes they're actually detrimental. But it just depends on what DNA is contained in that plasmid. Alright, and that's it for this section, guys. If you have any questions, let me know.